I'm Luke Marsden. I am employed through the Nansen Legacy project, so this is more or less 95% of what I do, work on this project. I'm employed for 3 years, set up at UNIS. I sit in the biology department, but my responsibilities span quite a lot further than that, as it's quite a multidisciplinary project. I work a lot with the geophysicists as well. A host of institutions across Norway. So I'll talk today for about maybe towards an hour. There isn't any formal exercise afterwards, but we can have an introduction to the tools and we can have a Q&A afterwards, and we'll see how it goes. So, yep, some credits before I start. So I've been working in this position since June, but before me, Paul Ellingson was the lead data manager in the project for the first couple of years. He now sits in Tromsø, some of you may know him. And he was instrumental at really developing a lot of these tools I'm going to be hosting. I've developed them a little bit further, but he deserves most of the credit. And then the data management part of the project is led by Øystein Goodøy at the Meteorological Institute and Tove Gabrielsen, who is in Kristiansand, I think. So today, I'll spend a few minutes quickly introducing the project. I think a lot of you probably are aware of little bits of it, but I'll go over the basics quite briefly. I'll talk about how we log data in the project, and since all the data in the project is openly available, or should and will be eventually openly available, I'll show you where exactly that is and how you can access that, as the data itself themselves might be of interest to some of you. We'll take probably a few minute break after that before we then dive in to introduce some of the tools that we have developed during this project that we hope can be continued and be useful in some other projects moving forwards. And as you can see on the top, we have some section headers so you can keep up to date with where we are. So on the right is a picture of that I took when I was on one of the cruises in February. I was lucky enough to go out into the Barents Sea. But this is like the backbone of the project, if you like. These cruises to the east of Svalbard, and there's 6 of them this year, although there's usually a few less than that. The project as a whole currently includes around 210 researchers across 10 institutions in Norway. There are some affiliated members who are further afield from that in different countries as well. It's a multidisciplinary project, so we include a lot of biologists, also oceanographers, meteorologists, some geologists, probably a few more people that I've missed there. And most of the work, although not all of it, takes part in the Northern Barents Sea, where we have multiple cruises that have been dating back to 2018, and I think the last one that is scheduled will be this year, although with COVID there is a chance that we may have extra cruises to miss upon lost time next year, but I think that's under decision making at the moment. And what's been important in this project, and what I will talk a lot about, is that everything we've developed from a data management point of view is committed to keeping the project aligned with the fair data management principles that are outlined by Wilkinson and a long list of colleagues in 2016. We've probably, for those of you who have been to a few of these sessions now, you've probably heard people talking about this in more or less every single one of these talks, so I'll make this brief, but we're now quite good at making data findable and accessible. This is usually a requirement for researchers when they publish papers, they have to deposit their data in a data repository. However, that doesn't alone mean that the data is interoperable and reusable. So that should mean that it's easily understandable, not just by humans, but by machines, with minimal manual intervention. And yeah, we'll talk about how the tools that we've developed help to keep people to those principles. So we'll talk about first how data are logged in the project. So we have a template generator that allows us to log samples and events electronically. This is one of the tools that we'll come back to later, so I won't talk too much about how this works at the moment. But basically, samples are labeled physically, but also logged electronically. And this has a distinct advantage that you can only fit so much information on a label. That's one thing. You can fit far much more extensive and useful metadata if you log it electronically. Labels deteriorate over time. Also, sometimes you might find a sample in a fridge several years after it was collected and have no idea what it is. But if you can scan it and tie that together with the electronic log, then you can find out exactly what that container includes. And we also include log any data where there has been no physical sample. This can be a sample that's been processed on the ship and disposed of. And it could also be something like a measurement of a physical property if we're recording the temperature of seawater or the wind speed or something like this. Everything is collected together and it's made available on SciOS, where we have a searchable database that's linked here. And every sample should be logged alongside a detailed sampling protocols document within that are published and can be found on SciOS also. These should be described or linked to within the database. And so if you ever want a more descriptive report style log of what exactly has been going on, then you can find that alongside any sample. This is also really important as it ensures that we have consistency between how we're sampling things between different cruises, where different personnel might be involved in working with the same data. So what is key to how we log data is that each sample is logged with its own ID. And that ID is not just unique within the project, but it should be universally unique. So we use UUIDs, which are 32-digit hex numbers. A hex number contains digits 0 to 9, but also the first 6 letters of the alphabet, which is then 16 digits, which is good from a data storage standpoint. And this is they look like this below, but this is quite a long, difficult thing if you want to type this out. So for simplicity, we can represent that as a data matrix, as you see on the right-hand side. And these have a incredibly small chance of collision. And there's different ways of creating them, but the way that we use for our samples is that the first, I think, 2 sections are related to the timestamp, and the rest are something to do with the network on which they are created. And actually you should be able to recover the timestamp by just having the UUID, although don't ask me how you do that. So that means that if you're creating UUIDs at different times, then there's 0 chance of collision. So we can print off these data matrix onto labels and print them onto the samples, and then we can scan them on again with a barcode reader. We also log data with a hierarchical approach. So each level of this data pyramid has its own UUID. So for example, if we visit a station at the top, we might have a few different deployments of different activities. Maybe there can be a CDT cast or a drone flight. There might be some extra instrumentation that's bolted to that. For example, sometimes you might have a Niskin bottle collects water attached to the CTD. And the CTD would then be the parent of the Niskin bottle. So this means that we can, if you have a whole suite of Niskin bottles, you can collect them together under the same CTD and understand exactly where everything is coming from and where everything fits together. And then you might have samples below that and subsamples. So this parent-child relationship is really important for us when we're trying to navigate through the database. Just some examples of how this might look. So if we visit a station called P2 NLEG04, we might have a CTD deployment and an MSS deployment. We might have an LADCP, master and slave bolted to that, along with a couple of Niskin bottles. So they would all have a common parent of the CTD. Then you might have some water samples from the Niskin bottles and some subsamples below that. But this can also work with more physical flights that are perhaps not as obvious, where you simply just have a single mission, like a drone flight or a dive mission, that's logged in exactly the same way, and any samples fall below that. And when it comes to searching for data, you can then not just search by the ID of the event itself, but also if you know the parent, you can then uniquely and reliably search for everything that falls below that. So this allows us to really get a good, robust way of quickly harvesting a subset of the data. The backbone of this project is this transect that we can see on the right-hand side. This is Svalbard on the left of this image. And we have 25 transect stations that you can see in yellow, and 7 of those are process stations. Now, these are frequently visited on this, and I guess the majority of our data comes from these stations. And it's hoped that these process stations in particular will be continued beyond this project in some capacity, so that in the future we can have a long-term log of data corresponding to the same or approximately same location. So these stations will hopefully be revisited frequently. But this is only, I guess, a small part of all the stations that we have visited in total. There's something around 500 stations in all. And for people who have been involved in large projects like this, having a reliable way of naming all your stations and things like this can be a little bit of a mess. But I won't touch on that too much. So we have different transects that are taken. For example, maybe we're looking for the polar front, and we might do a transect through that where there's multiple stations. We might deploy moorings or recover them at certain stations, and they'll have their own station as well. And then there'll be one-off deployments of different activities from time to time too. So it soon adds up, and whilst most of the data is collected around this transect, there's data from further west towards the west coast of Svalbard, and I believe some of this are going to be a cruise going quite a bit further north, up to 85 or 86 degrees or something like that as well. eventually everything is made searchable online, and this is a big part of my job. So when other researchers collect all their data, they send it to me, and I do some cleaning and keep this database up to date. So it's searchable on SciOS. It includes rich metadata along with that. So here, for example, you can see the ID of the sample and the parent as parent of that sample. We have information related to the date and time, where it's taken from, the depth of the water at a certain point, and who you should contact if you want to know more about this data. And obviously, this is also then linked to the physical sample. So this event ID is the same as the UUID, represented by a data matrix on the sample itself. So if you are wanting to find this, you can go to the SciOS page. Under Services and Data Management, we have a Nansen Legacy Tools option. And most of what we have is hosted under this location. This has been developed a little bit, but at the moment, so this interface will change a little bit, but I think the same idea will be brought forward. So I'll just go through this briefly. So you can search by a variety of different parameters relating to the date or the location, perhaps what gear has been used to collect the data, or looking at different types of samples that you might be interested in. You could also search by the station name if you have that, or the cruise number. And so, for example, you might want to search for all the CTD data where chlorophyll A has been recorded from the year 2018. So you can do that. And this will return a table like this, where you have a list of all the different samples. And each of these is a hyperlink, so you can, firstly, you can move forwards to the sample itself, but also you can look at all the, collectively look at all the children of this parent, for example, or look at all the samples with this type labeled onto them. When you navigate through to one of those events, you have something like this. You have a map showing the sample location, links to the parents and the children, so you can navigate through the database, and all the metadata that we have previously shown just a slide ago. There are other ways to search for the data as well. This is the main one. As I mentioned, some of this is being reviewed now as we're going through some development of the SciOS webpage, and hopefully this will be become user-friendly, but I think any of the useful features in this will be continued. So you can search by the ID of a sample. This is useful if you find the container and you want to find exactly what that is. You can look for multiple IDs if you have those listed. You can search for the station if you have perhaps the location, or the you want to find what cruise it was, the station was visited, you can do that. You can list all the stations as well. And it's even possible to output a full log of the data. What I should probably have mentioned already, is that this database at the moment is just the metadata, not the data themselves. So this provides a good overview of everything that has been collected within the data, but then there's usually a bit of a time lag between publishing this, which is done usually a month after the cruise, to when the researchers get round to finally publishing the data. But we can talk about the data themselves after this. So, as I mentioned, all the researchers are committed to publishing all their data in a fair way and making their data accessible via SciOS. We've had a little bit of a slow start for this, but I think it's really gaining momentum now, and some really useful data is now becoming available. So you can access this by going through access and data on the SIOS page. And this provides a single access point to not just Nansen Legacy data, but all data that is collected in and around Svalbard should be made available through this portal. And whilst no data is actually stored on SIOS, it instead links to the data repositories. And I'll show you a little bit how that works. So the data access portal looks like this. You can scan down, and all the Nansen Legacy data is assigned a tag AEN, that's Nansen. At the moment, you can see there's just 29 data sets published, but this is really gaining speed quickly, especially now researchers have a little bit more time to work on publishing their data. So if you click on this tag, you can return all of the data that has been deposited associated with this project. You return a map showing where all the samples have been taken with this included in these data sets. And if you scroll down, we have a table linking you to each of the different data sets. So you can either view the metadata themselves, which returns a table on SIOS of all the different discovery metadata that describes the data set and how to find them. Or you can click on this link, and this will do one of two things. Either it will download the data directly by clicking this link from the data repository, or it will transfer you through to the data repository and you can download it from there. So in this case, this data set is stored at NMDC, the data repository that IMR host, and it's possible to then download it straight from this platform here. We also, as I mentioned, have detailed sampling protocols that we publish and keep them up to date. They are also available on this Nansen Legacy Tools page. A little bit of background about these. These are a methodological agreement between the involved researchers for any given type of data that has been recorded. They're kept up to date. There's 6 versions currently on SIOS, but there's a 7th and an 8th, I believe, that are currently being published, and they will be made available when they're done so. And these are really important at ensuring continuity and comparable data throughout the 5-year sampling period. And they're pretty thorough as well. Most of the samples that have been taken within this project are documented along with detailed sampling protocols. They include, for example, a step-by-step how-to guide if you want to repeat the sampling protocols. So the same processes can be adopted by other projects or within the same project. And yeah, this just makes sure that if we have different people collecting the same data, you can rest assured that it's gone through some kind of similar processing and QC process before it's documented. And this should allow for easier cruise planning as well. If the cruise leader knows exactly, has an overview of what people are interested in within the project, then they can quickly scan through this where everything should all be documented together. So what has been really one of the biggest developments from a data management perspective in this project is the development of the template generator. This has been developed, was originally developed by GBIF Norway, although it was, I guess, in its infancy at this point, and was chosen after discussions with Dag Andersen. GBIF is a global biodiversity information facility. So for people who are not aware, they host a hell of a lot of biodiversity data, but also, I believe, ecological data as well. I think Matteo discussed them quite a lot in his last presentation. This template, whilst it was developed originally by GBIF Norway, was heavily developed by Paul, my predecessor, and he's done a lot of work to make this specialized for multidisciplinary data, as like this project is. So whilst the bulk of this is based on Darwin Core, there's also fields that are taken from NetCDF-CF conventions, which are more suitable for physical data, for atmospheric data, or oceanography data. And there's also some other terms in there that are perhaps more specialized to our project. The advantage of having these templates is they allow us to generate standardized templates where we are sure that everyone using them is adhering to a common vocabulary. And that means that if you generated a template with certain column headers, you have a description when you're choosing which headers to use. So you are clear what should be included in that header, in that column, but also someone then reading the data later will also be able to have access to that description, and will then there'll be no confusion about what is included in a certain column. This is continuously developed and added to. In fact, we're probably going to go through quite a big round of development this year as we're trying to get a better overview of all of the data that has been recorded in the project, and making sure everything is encapsulated by this. I'll briefly touch upon Darwin Core and NetCDF, as there have been previous trainings on this. But in brief, Darwin Core focuses mainly on primary biodiversity data, for example, occurrences of species. But more recently, it's been developed for ecological data, like descriptors or traits. For example, you might be looking at the length of different fish or something obscure like that. The main part of Darwin Core is a table or a CSV file that has column names that are taken from a common vocabulary. And this template generator will allow us to generate exactly this. NetCDF was originally developed by atmospheric modelers, but it's been developed to include oceanography and meteorology and some other disciplines interested in physical data. And similar to Darwin Core, all the standard names are searchable online with descriptions and units for what they should be comply with. I'll talk mainly about Darwin Core for the remainder of this, where, as I think this is probably more easily compatible with biodiversity data, or I'll touch a bit on how it can be compatible with the NetCDF, creating NetCDF files afterwards. So Darwin Core archive includes a core file, which is columns of data recorded one row per record. This could be an occurrence core where you log different occurrences of every time you spot different species. It could be a taxon core where you log different taxa that you spot at a certain location. There's a few different examples. And this can also be surrounded by extension files, which allow you to have a many-to-one relationship with any row in the core file. So if you have an ID for a row in the core file, then you can refer to that here in the extension file. So if you have, for example, if you're logging in the core file occurrences of a certain species, you might want to have an extension file that has the scientific name for that in different languages or something like this in the extension files. Hello? Someone speaking? No. Then we also have a metadata file, the meta.xml file. This describes the structure of the data. So it describes exactly how the core and the extension files are laid out and how they talk to each other. And you have an eml.xml file. And this describes the data set as a whole. So this is like global attributes, if you like. Things like the abstract, who's involved in the project, any methods that have been carried out, citations, maybe where the data has been collected, things like that. The archive itself is just all of this collected together in one folder and zipped together in a zip folder. And how does this relate to what we're doing and talking about today? Well, I'll show you how both the core file and the extension file can be created using this template generator. And then I'll talk about how the metadata file and the EML file afterwards can also be created quite a lot simpler, having used these templates to create the core and the extension. So again, to access the template generator, you can go through the Nansen Legacy tools. So on SciOS, go through Services, Data Management, and Nansen Legacy tools. And we have here the Excel template generator. So if we click on that, and this is what it looks like at the moment. Whoops. At the moment, what you can see is we're in the Nansen Legacy setup on the top right, AEN, but there's other setups that we can choose. I'll talk about those later. And we have all the different sections subdivided into sensible categories. In Nansen Legacy, we have certain required terms. We require each of our researchers, when they're creating one of these logs, to include. We also have recommended files, which we suggest that they include, although they don't have to. And this extends quite a lot further down. You can see just perhaps 20 or 30 here, but there's probably hundreds of different selections relating to different parameters through biology, oceanography, meteorology, and this has been expanded over time. What I'll talk today is if we switch this to the Darwin core setup here. So in this case, each of these different boxes represents different Darwin core classes, which corresponds to different classes on the Darwin core website. And each of these terms is a term within that class. And as we can see, there's more different classes down here. So event classes, location, identification, organism, and a host of different terms as well. If we look closer, we have different languages we can choose. So English, Spanish, and Chinese. This doesn't change the language of the generator page, but it changes the language of the template that is created. And on the right-hand side, so if you hover over one of these fields, So for example, we're hovering over event time now. We can see the name of the standard name in square brackets. We have a description of the field, describing exactly what should be included in that and the format it should follow or any conventions. And we have some formatting requirements here. So it should be UTC time in hour, minute. And what happens if the minutes are over 59, and the hour will automatically be added a value of 1 to. And so we have a readable term here, event time, in more of a readable format. And then this corresponds to the Darwin core term in the square brackets. So we just go through the different sections and select whichever boxes we want to include in our template. When we're happy with that, we can select Create Template at the top. And this will open directly into Excel. And it looks like this. So what we see is that we have a few different rows. We have the more readable row in the second row here, and we have a third row which corresponds to the Darwin core term. Now this is usually hidden for good reason. This is protected. It will be a lot easier to read the data in later if wording of this is correct, and therefore if there's a typo in here, it can make your life a little bit more difficult. And then one row should be for one event or occurrence within this. The notes that we saw on the right-hand side of the template generator are also left stored as notes whenever you select a certain cell. And this is really useful. For example, you might be given a template by your supervisor or by a colleague, and you don't then have to revert back to the template generator. Everything that you should need to know about how to fill in the template should be all on the same sheet. So how can you create a Darwin core archive from this? Well, it's possible to do this through the GBIF integrated publishing toolkit. I believe that Matteo covered this briefly in a previous training and in the fourth training of this series. So his slides are available on SciOS if you want to refer to those. What I'm going to go through now is how specifically using this template to generate your file. The template generator to generate this file will make it hopefully a lot quicker and easier to create this archive. So let's say we go to the template generator and we're taking the default setup at the moment. We've selected some of these different column headers that we have selected, highlighted in red here. We generate our file and we fill that in. In this case, we're looking at different occurrences of Orangitha, Tarandus, Platir, don't make me say that, but basically Svalbard reindeer, all observed on successive days in April in Longyearbyen, so 78 degrees, 15 degrees east. Once we've filled all that in, we can have a look at the third column, the third row. I've unhidden this. This step is somewhat optional because you can specify how many header rows you have, but I think it's neater to just then remove the first two columns so it is containing only the Darwin core terms. I think this will be more easily understandable to someone who is finding the data and who is less familiar with the template generator but might know all of these, be familiar with these Darwin core terms. Then we can just save that file as a CSV file. So occurrences.csv in this case. And if we go to the IPT tool, so this is integrated publishing tool, you can decide that now you want to give it a short name and create a new resource. It's just a test one with an occurrence file. So I hit create here. And you can choose the file that you've just created using the template as our source data. So we find that. And we've uploaded that file here. The input file name is listed here. We can use this information to describe the formatting, but this all looks okay. It's comma separated. There's one header row. And we can have a look at the source file. Okay, there's 11 columns, that's right. Certain number of rows, and we can analyze that by just selecting this analyze next to the eye icon. And we see that this looks as we would expect it to. So if that's all being read in correctly, so we have our source file, now we need to map that. And this is where having the template generator really comes into fruition. So what the mapping does is automatically map, well, automatically or you manually map the column headers you already have to the Darwin core terms. Well, because we've used a template generator, all of these terms should already exist and already be able to be automatically mapped. So let's try that. So we hit add here. We have a created source file. Yep. The mappings. And remember, we had 11 columns here in our source file. So when we hit the mappings, we have to make sure we select the right source data. We only have one in this case, so that's fine. Just hit save. And we can see 11 of the columns have been auto-mapped based on their column headers. So that's done most of the work for us. We just have to check. So we can scan down. On the left is the Darwin core terms. And here is the different column headers that we might have. You can select from a drop-down box. And we can see that this matches up, so we're okay in this case. Right, so that was quite simple. Was there anything else I was supposed to say on this? Nope. 11 terms are mapped to the occurrences. Great. So now we can go on to the next bit, which is adding the metadata files. So this is then the file to describe the dataset as a whole. So we hit edit here. And we just go through filling in all the different boxes in this case. This can be quite long-winded depending on how much metadata you want to include. But you can go through here, press next or save, and it will take you through to the next section. And it's just like filling in any other form. And this will then automatically generate the EML.xml file from this information. Okay, so after we've done that, we have the metadata file now created, and we can just go ahead, preview this file if we like, and we can even use IPT to directly publish this data onto GBIF. So just to review that, so the core file and in some cases the extension files too can be generated by the template generator. You can then much more easily map them to the Darwin core terms if they've already been selected. And the meta.xml file and the eml.xml file will be created by just filling in some basic information on IPT. And then you can just automatically create this archive and publish it all in the same tool. I'll talk just very briefly about NetCDF, as it's a little bit more of a complicated format. As there's more dimensions and possible scenarios. We've also covered how to create CDF files in a couple of different trainings. We have had the how to use Rosetta to create to go from a tabular CSV file, such as this one, into NetCDF. That was given by Toril in session 3. And Eystein covered how you can do this in Python and R in session 2. But if you ever need any help with any of this, just contact your favorite SIOS employee. Yep, so how this relates to the tool that we have set up is that a lot of these physical parameters that you might want to include on the recommended terms within NetCDF should also be included as column headers. This isn't a fully exhaustive list at the moment, and as I say, this is being updated all the time. But please, if you do ever have any suggestions for other things that could be included, or anything, or any way that this could be improved to make this transfer to NetCDF a little bit simpler, then just get in touch with me. And one tool that's often overlooked is all the code that we have set up to set up this system in our project. And we know that similar setups are now being used in a couple of different projects, mainly by Nansen Legacy participants who have work commitments elsewhere. But I think some of these tools, like the template generator in particular, but also hopefully some of the logging systems will be continued in different projects in the future. So all of this is open source. It's all available on the SIOS GitHub page. And if you ever want to adopt a similar setup for your project, just get in touch with me and I can help you try and get that working with you to suit your needs. And that's everything that I had to say today. So I don't have a formal exercise for you, but if you would like to spend some time exploring the template generator and what you can do with that, or if you want to look through the metadata catalog, I will stay around for a while and answer any questions that you might have. Thanks very much for listening. The poll is now on there, so if you